So to end the session, it's my pleasure to invite Professor Adam Cohen from the Netherlands to come and deliver the final keynote lecture of the day. Adam is a graduate in both pharmacy and medicine. He did much of his early training in England, first at the Wellcome Research Laboratories and then King's College Hospital. He's now Director of the Centre for Human Drug Research and Professor of Clinical Pharmacology at Leiden in the Netherlands. He's also been the European Editor of the British Journal of Clinical Pharmacology and is a member of the Council of the Royal Academy of Science of the Netherlands. Adam.
Thanks very much, Simon. And while studying the program, I started to detect a sort of rather frightening sort of pattern. Is that you suddenly, when you were just doing experiments and producing results, you never had to give a keynote lecture, and then suddenly there comes a day where they ask you to give a keynote lecture. And I noticed another pattern, and that is that all my predecessors got about half an hour for their keynote lecture, and I get a full hour. So this, apparently the pattern is that it gets longer and longer and longer until you have unlimited time for your keynote lecture, and that moment is getting frighteningly near now. I'll try to keep you not too long off your drinks. And what I'll try to do now in the next hour is not really to give you information, but to give you a couple of ways of thinking about drug development. This is not about prescribing, and the reason is my talk was moved, but I hope you can make the mental shift with me. And these funny words like dream-driven drug development and monodimensionalism, I'll come to, so don't worry. This is the Netherlands on a good day. And this is going the wrong way, which is interesting. I'll do that again. Okay, well, this is one of those famous slides that you shouldn't be able to read, but it's the map of the all the mediators involved in Alzheimer's disease. It's done with a new language, which allows you to put all the literature in one of these schemes. It's obviously frightfully busy. And it's all things you can influence using drugs. You can inhibit them or stimulate them or block them, whatever you like. And in fact, if you look through what is happening in biotechnology, it's probably true that somewhere there is a company doing any of these sort of things and blocking them and getting venture capital to do so, which is fine, but the question is, how do you deal with this? And when people are still talking about a pipeline in a drug company, when it's sort of image of where something goes in at one end and everything that goes in comes out at the other end, then that is also getting slightly funny. Of course, they leak. And the question is, in drug development, how you deal with making these things leak early and only the right things coming out. It's not a pipeline. The biology is becoming, and the genomics and the genetics are making the biology frightfully complicated. And as clinical pharmacologists and drug developers, we have to do something about this. And it's not entirely obvious how to do that. Now, just to give you a horror story we recently dealt with, this is a sort of, these are doses of a drug, and this was a noradrenaline reuptake inhibitor invented by in fact, Australian company from the neurotoxins of the cone snail, a very beautiful thing that lives on the Great Barrier Reef. And this had to be given intrathecally and should be a drug for pain. And this is how they sort of developed it. They gave it to rats and then to humans, and they increased the dose in humans because that's what people learn when they do new drugs. They say you give this until a tolerability level, and yes, they found the tolerability level. It was about 40 milligrams. There was a patient, a cancer patient, who had a seizure. Quite to be expected if you inject a noradrenaline reuptake blocker into the central nervous system. Well, they found the level at which this happens. It wasn't very useful. And then they from that moment onwards, they spent 5 years going through all these doses in different species in humans and testing and FDA holds, which they had to recover from. And in the 5 years, they spent $50 million. And at that point, they came to us, and we asked them the question, did you ever look at this drug actually treating pain? And they said, no, we didn't get that far. We were just dealing with the tolerability of this compound. So we did it. And, well, this is one of those really simple pain models you can apply to a group of healthy volunteers. It's a bit fiddly because you have to put a catheter in the CSF to inject a drug, but it doesn't take very long. It takes about four weeks, and it's not very expensive. And these are the different doses of the drugs and placebo, and you can see them overlap, and only with a slightly higher dose, but it was a very small effect. Not really worth putting a drug in somebody's back. Very interesting question why this hadn't been done first, and company is now out of business. And this is not uncommon. I'm not saying it happens every day, but this sort of thing happens quite a lot as people are trying to deal with all these different ways of affecting biology. Now, at the same time, there is an industry that is in a bit of trouble, right? The pharmaceutical industry is not exactly popular. It's continuously thinking about how to save money. And it's funny because on the other side there's this huge, rich biology which you could explore. And of course it's an industry that started with mainly chemical innovation. This is a word I'd like you to remember. They were producing molecules. This is how it started. The pharmaceutical industry started producing molecules to dye garments with, and so they became sulfonamides. Worked really well. Especially in the 90s when they discovered you could sell this at a really high price. Now, if you sort of multiply the two, right, you got something new with a patent, and it's a very high price. They then started to call this blockbusters, and this is a lot of money coming in. And it was at the time of the cholesterol-lowering agents, the ACE inhibitors, really quite safe drugs, where the dose didn't matter so much, and there were wide populations benefiting from them. Could this go on? No, it didn't. Because, of course, immediately what happened is that the companies started to produce, what we sometimes call Me Too drugs, but incremental innovations, still to be sold at a high price. And it became much less scientifically interesting. So in my career, the very strong integration of pharmaceutical industry with academia deteriorated, disintegrated, and it seriously affected recruitment of talent. I was working for Jim Black in a time when Wellcome still had 4 Nobel Prize winners in the staff. Now, I'm not saying that is the answer to everything, but the scientific structure of these companies changed quite a lot. What also happened is because of the high prices, the access problem seriously affected the social reputation of the industry. That wasn't the case 30 years ago. And if you now go, and this was in India where I was, it's really a remote village. And if you walk through the village, you see advertisements for mobile phones everywhere. You can actually charge your mobile phone. There's no electricity in the village. You can charge your mobile phone. High-tech product being distributed widely throughout a very poor area. And if you go to the doctor there, then it's fairly clear that you won't get penicillin if you have pneumococcal pneumonia. You can't easily get it. So the access problem of drugs has become a problem. It is now also a problem in the developed world. If at the renal unit we discuss eculizumab for complement-induced diseases, we talk about 500,000 euros per year we have to spend on a patient, and there is a serious access problem in a country as rich as the Netherlands. So that is one of the things that happened. Because of the drugs being so expensive, and this is something that mainly bothered the industry itself, you got a generics industry. It's actually quite strange that they exist, if you think about it, because most supermarkets, when they had something that other supermarkets produced, would just lower their price until the other supermarkets were out of business. But it has reduced the commercial life a lot because the industry kept doing their thing with price. Now, of course, you've got price controls. This is something managements of drug companies are continuously dealing with, trying to increase prices, and governments are trying to reduce prices, and this goes on forever. And there's all sorts of controls being done to sort it out. And it made the whole system extremely risk averse. Things that are so expensive have to be very safe. So I think that a large part of what we have in the regulatory expansion, the explosion, some people would say, is induced by still these high prices. And then finally, the incremental innovation, which was by far the easiest way of dealing with blockbusters, right? It's just lowest resistance route is to produce incremental innovation rather than totally new drugs. It's less interesting to clinicians, and you got something which we never had. You have this sort of competition for patients and sites when people were all developing the same cholesterol-lowering agent. And this had also serious effects on development time. There is no doubt that that sort of thing also folds back on itself. And that in itself made costs rise. And then finally, well, I'm pushing the wrong button, that's what I'm doing. Finally, of course, the industry responded in the last 20 years by getting bigger and bigger and bigger, because that is what they learned in business school, and larger companies have a larger market share, and therefore they normally get more profit. They also increased the marketing efforts for drugs. It's particularly interesting that a British company producing antidepressants got the biggest fine ever for sort of bad marketing practice of $3 billion in the US. It was in the same year that the chief executive of that company said that they would go out of research in antidepressants because of the fact that they couldn't have good outcome parameters for it. And then you automatically start to wonder what would have happened if somebody would have put $3 billion in research in genetic or phenotypic outcome parameters in depression. And what it's led to is political pressure on governments and the other way. In other words, quite a lot of sort of friction energy going in the wrong direction, ending up in all this going back into this vicious circle, not getting anywhere. And you end up with a fairly unpopular industry in a highly regulated environment, under price pressure, with shortened commercial life. And this is about what we're seeing at the moment. And this is funny, because there's quite a lot of work to be done. My Alzheimer map you could replace by any other map for any other disease. We're not doing too well in many of these diseases, and we really need the power the pharmaceutical industry has to do good research. And I think in future, this is a very, very important goal to reach, is to actually get that friction energy that we, for some reason, generated in the last 20 years into research energy. In other words, not pay $3 billion to the American government, but get $3 billion, apparently it's there, into research systems, because it's really necessary. Now, what has happened up till now, I think, is what I call monodimensional solutions. If you look through the field, you always see somebody somewhere who's got the solution for all this. And it's generally one thing. So I'll show you a couple of these things. When we talk to biotechnology companies, they all, as one man say, to get effective drug development, what we need to do is we need to give a drug to people and check if it's tolerable and safe in phase 1. This is apparently something that most people agree with when you say it. Is that right? Well, I don't think it is. It's just a solution for nothing. You've just seen where it goes wrong. This is somebody, it's probably unrecognizable. It's me in the Wellcome Research Laboratories doing a vascular experiment. And we, as a matter of course, in Wellcome, where we had our own unit, did pharmacodynamics in the first administration to humans. This was lamotrigine, Lamictal, which I got to give to people for the first time in the world. And we in the first trial, we did experiments to show that it produced less ataxia than phenytoin. And this was very important for advancing the drug, and eventually it sold, I think, £1 billion or something for GSK. But the link between pharmacology and sort of toxicology is gone entirely from the field. And it is, again, quite interesting why that happened, because it's so logical that when you give a drug for the first time to humans, you also check if it works, if that's at all possible. But it's very rare. And so within all these systems, this sort of solution has disappeared. There are also people who say that what you should do is reduce the rules, get rid of the rules, harmonize the rules, whatever. And at the moment, there's whole groups of people working in Brussels, and I've actually been part of it for some time, on a general Europe-wide regulation for drug research, which apparently is going to solve all problems by making it all fast and efficient. And in fact, it's the wrong rule. No one notices that, but it's the wrong rule, because if you look at the research output of all the countries in Europe, it's so varied. You know, it's in the level it comes out, and if you look here, there's the number of publications per thousand population. I show you this because the Netherlands is the highest, of course. And we could also divide it by other things, and number of pigs in the country or something. Then the Netherlands would be even better. But it is generally a group of countries, Belgium, the UK, Austria, Denmark, and the Netherlands, that lead this. But at the other end of the spectrum, there are, of course, countries in Europe which are treated equally within this system who have very low research output. So to make one rule for this and to try to harmonize, you know, approval times, which in our country are very short, and if you go to Czechia, they're very long, would be quite difficult because it's totally different research. Same goes for the time it takes to start patients, a huge variety, which you can't actually reduce by changing the rules. You have to reduce that by bringing the people who are not so good up to the level of the people who are good. And what we're trying in Europe is to get back to the average, which means that the countries at the top will be brought down and the others probably won't go up. So in a country, in a part of the world where we still have countries like Italy with 1,000 ethics committees, and some other countries like the Netherlands with only 20, and that's you first have to take care of that, and that will take before you start taking the rules. So the monodimensional solution, which is in the newspapers and in the journals quite a lot by the people who make these solutions, that this will solve anything is ridiculous. Same thing, and we show that it is actually not the rules. It is actually how you interpret the rules. We've written a paper about this in the British Journal, where we did evidence-based data monitoring for clinical trials. We just looked at what data monitors find when they look at clinical trial data, and we tried to see if any of these things they find had anything to do with the result of the trial. And this is what we found. You have about, these are called queries. This means somebody goes into a plane, flies to the Netherlands armed with an enormous amount of post-it stickers, and sticks post-it stickers into data forms. And they do this about 1,400 times. And then we had an independent committee grade if actually these things had anything to do with a change of the data point. Well, that's only 4% of the cases. And then if there was a change, did it have anything remotely, we were very strict about this, to do with the result of the trial. And this happened in 6 cases. It's about 0.5%. And these were things like the attribution of a side effect not being probable but possible or something, but we were quite conservative about it. So here's months of work for lots of people, not actually yielding a single bit of useful information. And that has nothing to do with the rules. This is just the way how people interpret the rules. So I think we'd be much better off by being sensible and data-driven about rules rather than trying to change them in Brussels. So it's not a very good solution. And what we said in our editorial in The Lancet, which we wrote about it, is that you shouldn't fix the roof of a building where the foundations are shaky. And the whole foundations of this are, of course, shaky because we are not going to work in the next 20 years only with drugs. Treatment of patients is going to be mixes of surgery, drugs, nutrition, devices that produce drugs, food that has an effect on drugs and medicines. So to have a rule system for medicines only is referring back to the sort of 1930s, where drug companies were still making molecules. And the problem is, of course, we have to try to understand diseases. And to have a rule system to get molecules onto the market is about 20 years out of date. We're doing it because we're monodimensionalist about it, and we think that it helps. The other things where we thought that it would help, the drug development from being integrated has gone, like many production processes, into what's happening to your iPad when it's being produced. It's offshored, it's outsourced, and it's done by very limited, specialized companies. What you just saw, the 50 million disaster of this Australian company, was a sort of conspiracy of many extremely reputable and well-meaning organizations who were in one of these offshore systems and didn't work together very well. It cost quite a lot of money. And the archetype of this sort of disaster was, of course, TGN 1412, which we described in The Lancet as a sort of paragon of disintegration of knowledge, which eventually led to a group of young people getting about 500 times the dose they should have had. And this was easily calculable if somebody would have integrated the whole thing. So you can't offshore a complicated process, and you can't outsource it, and we're slowly learning this. But if you look back when this started, you see people giving monodimensionalist solutions and say, look, this is all a question of cost. We have to make it cheaper by doing it in a cheaper country. And if we have lots of these specialized organizations, then they all can concentrate, they can focus. The word management consultants always use the word focus. They can focus on something and they will do it better. But they don't talk to each other, so then you get quite serious problems. We wrote a piece about that, Jim and I, in the British Journal on the industrialized research, and we used neo-Luddite. You know, Ned Ludd, who was the guy who smashed the looms of the Industrial Revolution because they were artisans and they didn't want this to be mechanized, and we felt a bit like that, so we read it and you can see what we mean. In fact, JeanPierre Garnier, the executive in pharmaceutical industry, leading Novartis, said it after he retired from Novartis. R&D is not scalable. You can't industrialize it, and you can't just drive it by metrics. But the monodimensionalists have been doing that, and the result is loss of passion, and I'll come back to passion, and loss of transparency, and loss of accountability, because everyone always has another organization that does this for them. So that's another bad outcome of monodimensionalism. And I think what we need to go back to is what I sometimes call IQDD. It's intelligent drug development where intelligent people who are well trained think about how to do it. They do at the moment, but they do only part of it. And this is one example. Of course, we did it. I'm sorry. We're not always intelligent, but in this case, we try to be. It was the first in human study with a new hypnotic. So we did what we call data-intensive studies, where we collect lots and lots of pharmacodynamic data when we give drugs to humans for the first time. There's an orexin antagonist, a drug that works a bit like giving somebody narcolepsy. And we did, at the same time, within the first administration to humans, a positive control, because that allows you to see how a drug relates to what's already on the market. And then we use techniques which are very old. You have to have these techniques for a long time to be able to use them. And then you can, this is adaptive tracking. It's a test developed by the Royal Air Force around the Falklands War to test for residual effects of hypnotics, very sensitive. And if you give 1,000 milligrams of a drug, you get this sort of decrease and increase in sleepiness. And this is zolpidem here, which works shorter, has an effect that is slightly smaller. And if you wanted a zolpidem-like bit more long-acting dose, you give 400 milligrams, and that's what they did. There's some other, you can do lots of tests, and you see about, they're totally different tests. This is postural stability. You can just see that the results are the same. And you can think of this as an analogy of any other drug. This is CNS, but we do exactly the same for inflammation by doing ex vivo lymphocyte stimulation, and you can immediately see the size of your effect. Now that sounds almost too obvious to tell to a group of people like yourselves. But it is fairly rare in drug development at the moment because it's been disintegrated. It's not integrated. There are organizations who can give drugs to humans the first time extremely well, as long as they don't have to measure anything. They just take out the blood for the pharmacokinetics. It led to a paper in Nature Medicine. It led to Actelion selling the drug to GSK for quite a lot of money. Eventually, and this also shows the weakness of these systems, the drug ran into a liver toxicity problem late in development and was discontinued. But that is still the inevitable risks drug development has, and which we still have to solve using other forms of biomarkers. So the problem is multidimensional in general. The solutions are multidimensional, but the problem is also multidimensional because we have to treat different compounds in different ways. And I think about innovation not as a new molecule, something that has something stuck to the side chain. So it's now a new molecule, you get a patent for it. But what it is chemical innovation. If it's still a cholesterol-lowering agent with exactly the same mechanism as the previous one, it's chemical innovation. We got 5 or 6 of those. There's, of course, also therapeutic innovation. You can have the same drug which you use in another form. Thalidomide is a good example. Or you could have mechanistic innovation where you use an entirely new mechanism to treat a disease. You also do that with a new molecule generally, but it could be even a new mechanism found for an old molecule. So here you got about 8 different forms of innovation, and they need different ways of dealing with it. Of course, the sort of generics corner here at the back, it's not therapeutic innovative, it's not chemically innovative, it's not mechanistic innovative, it's just a new ibuprofen tablet. It needs a totally different environment to deal with it. It's totally mechanistic what you have to do. You don't have to think about it. Then the high innovation corner where you got a new molecule, a new mechanism, and a new therapeutic field. So this is, I think, where most of us like to be, I think. But it's important to recognize that other corners exist, and you have to behave differently. That, I think, is something you occasionally need to explain to people. It's not only chemical innovation that drives our field, it is much more than that. So this sort of thing is important to realize. And what we're going to see, of course, is this all being even worse with targeted therapy. Not only one drug, but several drugs. You can treat melanoma extremely well with a BRAF inhibitor for about half a year, and then it comes back. So you need to have 2 drugs. This makes the whole field obviously at least twice as complicated. And then we have drugs that come out of devices, drugs that combine with nutrition. And so the future for anyone who is still in the stage that they don't have to give keynote lectures is actually quite nice because you can deal with all this. But we have to deal with it in a different way than we're doing at the moment. And so that means getting rid of monodimensionalism. So I give you one other thought idea, is what I call prototype development. It is important when you're in drug development to recognize that you deal with a prototype. And we think that you deal with a prototype if you don't know the link between the molecular mechanism and the clinical effect. So it's not a beta blocker, there we know, but it could be a toll-like receptor 7 antagonist. We know it inhibits the toll-like receptor 7 or 8, or 2, or 3, they're all there. And we don't know if that links to improvement in psoriasis. So that is the sort of one axis. The other one is that you may not, it would be preferable if you had a marker for the link. In the case of psoriasis at the moment, we just take pictures of the patients and rate the amount of lesions they have, and then at the end we do some image analysis. But it would be a lot nicer if we could see something happen in a skin biopsy, and if you have that, drug development becomes easier. So drug development today is going to be moving from prototypes to generic compounds by first biomarkers and innovative trial design, and you have to do both. You can take a huge leap. You can do a trial, large trial in a group of patients, and hope that you're right, but of course you normally have the wrong dose if you go on the wrong dosage schedule or you got the wrong patients, so you won't get there. You see biotech-driven companies quite often do that. They got no money or they haven't got enough money, so they take this huge leap into the unknown, hoping that their prototype will work. And you only have to read the sort of journals, the journals about the life of a biotech company, and you can see that that often goes wrong. So a standard compound, if you wanted to recognize it, is a product. You know, it could be another antibiotic. And it's very easy to deal with this uncertainty, so they're generally quite simple. And it's easy to work with stage gauge. This is where you can use phase 1, 2, 3, and 4, as we all learned. It's possible to first finish a package of work, and you call it phase 1, and then go to the next package of work, and you call it phase 2. Because you already know which trajectory you have to take through getting the knowledge. And so it's linear, it's confirmatory, it can be done very fast and very cheap. So this is the sort of stuff you outsource and offshore and use. And it can be industrialized by differentiating and disintegrating, like you built your iPad, you design it in San Francisco and have it built in a rather badly ventilated factory somewhere other country. I don't think it's great fun to do this, of course. No one in a scientific environment likes to develop these drugs. It's actually rather boring work, and I think most scientists much more would like to deal with prototypes, which are concepts. If you find something and it's negative, you've still shown something. If you've shown that the toll-like receptor 7 has nothing to do with psoriasis, this is still important scientifically. It's nice because there's so many uncertainties and you can redesign the whole thing to reduce them. It's cyclical, which means that you test and you go back to the drawing board, so there's much more interaction in the prototypes between different parts of this system, and that is, I think, why you now see drug companies move back in with universities, because they need that interaction, and it'll be great fun to manage it. Information becomes much more important and knowledge becomes much more important than speed. Just look at my initial reuptake inhibitor example. If somebody had said, or TGN-1412, if somebody had sat down over a cup of coffee and thought about it very carefully, all of this wouldn't have happened, or possibly wouldn't have happened. So it's learning research rather than confirmative research, and it's integrated. You can only do it if you put specialists in one environment from different disciplines. It's different. You can't do this by email. You have to do it over coffee and meat. And this is how all big companies deal with their prototypes. And if you read Steve Jobs' biography, you'll see that is exactly what he did. Is put all these people together and get involved in all the different parts of one of those products. So it's concurrent engineering. It's not in sequence, but it's together. And you can't easily do technology transfer. Apple or any other innovative electronics company will never technology transfer their prototypes. They do it when they got the whole thing worked out, and then they can have it produced cheaply. So it happens in networks. And I think most people who are in academic environments recognize that they have all the components in any big university hospital or group of university hospitals to do this. And it's interesting that we don't. And it's just something to think about. Prototype development. We've written a paper in Nature Drug Development Reviews, which you can read yourself. I think this is one of the thought schemes in drug development which don't do this in sequence, but do the whole process as an integrated way. So normally you got animal people and they do something in animals, and you got human people who do something in humans. But of course it is related. So you can take the animal receptor and try to relate it to the human receptor through different species or different techniques. Happens in drug development, but very rarely do you see somebody who's actually made this, what we call translational link. It just happens, if you look at these dossiers, which I've done a lot, it just happens in a different section of the dossier. You can read the Ka at an animal receptor, but it's rarely put together. Same goes for cell effects, metabolic effects in cells, or tissue physiological system effects. And finally, of course, whole organism studies. And then the human studies become, in fact, a reflection of this, the preclinical, which is also put in time. You first do preclinical, it is true, it's because of safety, and then you do clinical work, but it can still be a reflection of this, what we call translational process. So then the human studies are not phase 1, 2, 3, 4, but they're studies that cover human pharmacology, or they explore a therapeutic effect, or they confirm a therapeutic effect, or they eventually get out in the field and see how the drug behaves in common use. But to be able to think that throughout the development you're still looking at how these things all hang together, I think is fairly uncommon. And if you look at the average investigational medical product dossier that's handed into a regulatory authority or a drug company, or from a drug company or to an ethics committee, they're generally extremely disjointed. There's no common sort of integrating paragraph written in it. And that, of course, leads to what we call question-based development. And this is, again, you'll find it in the paper, and then we give more explanation, but it's a way of thinking about objectives, because all of us may be in a situation where we get a new compound, it's only been given to animals, and somebody asks you, write a program. Well, you can do it in two ways. You get a book on drug development and you look at the chapter phase 1, you copy the objectives from that chapter into your protocol. That's what most people do, so you're in good company, and then do the study. Of course, you don't get any information. What we try to do is make people think about these sort of 5 large groups of questions. Does the drug get to the site of action? Right? Some people call that pharmacokinetics, but we don't. Pharmacokinetics is only there because it tells you that the drug is around and for how long it is around, and that it actually crosses barriers. And when you have a question like that in front of you with this sort of mindset, you are actually going to think about biomarkers at the same time. Because if you think about, you don't think, I'm going to do a pharmacokinetic study. Yes, you get the half-life, or you get the absorption half-life or something we all learned. But you're actually thinking about, does it get to the site of action? Where is the site of action? Where do I think it is? And maybe it's the brain. So maybe you have to do something and find a biomarker to see it gets into the brain, rather than, and we've got examples of that as well, very good willing people who develop a drug for schizophrenia that never got into the brain, and you can go on doing that for quite a long time because it produces no central side effects, which is quite nice, but it also doesn't work. So same goes for pharmacology. People always want a drug to work for a disease, but we feel that first you should show, if you can, that the drug has its pharmacology, the intended pharmacology, that it doesn't, that it's not about, I don't know, twofold above its maximum effect on a certain receptor system. Does that make you better? No, it doesn't, but it is a prerequisite for doing that. So now a pharmacology study becomes something that is integrated in a thinking process around the development of drugs. And then finally, clinical is of course, in fact, first, do we affect the pathophysiology? And again, there are many diseases where you can't do this, but the number of diseases where you can actually study the pathophysiology quantitatively is increasing enormously because of all the techniques we have. So you can use those, and if you're lucky, you can study this, and that is the link between the etiology, which if you know it, it becomes a lot easier, and disease modification. That finally brings you to the question, does the patient get better? And you can say the same about adverse events. I learned from Jeff Aronson that adverse events aren't magic, they are just pharmacology, and they are. So you can study them pharmacologically. And even if they happen incidentally, they still mean that there's a mechanism behind them. This gives you a therapeutic window, and finally you see if there is a population of patients in which all of this happens. So that's question-based drug development. So you set up a set of questions, and you tell which biomarker you use to answer the question. If you have a question that can't be answered, that is a risk. That's a development risk, because you can only answer it in the end by using the drug in practice. And so once you've got all these things, the ascending first-in-man dose study becomes something totally different, because the way these things normally go is that you start at a low dose, and then you increase the dose, and you increase the dose, and you increase the dose, and this is where you reach a maximally tolerated effect. This is where the cancer patient has a seizure, or somebody vomits, or whatever. This is how William Withering developed digoxin in 1785. And then you don't know where the drug works, because this one you didn't measure, and this one you didn't measure either. So if you would, with a drug with a small therapeutic margin, like digoxin, these things are close together. So actually, and this is why we do it, if you give a cytostatic to human beings and you say the maximally tolerated level is where your lymphocytes are going to go down, you actually are fairly close to where it probably also will work, because it's a rapidly growing tissue and you inhibit it, but it's a rather primitive way of finding it. All of the literature why we do phase 1 studies like this is based on cytostatics. What we got now is very selective drugs with very high therapeutic margin, and so it's necessary to measure a pharmacologically effective level, because that's much more likely to be close to the effective level, which you sometimes have to take 10 years to find, than the maximally tolerated level. This is just human toxicology, and we don't have to do that. So this is, again, new ways of thinking about these studies that are done so often without these things still. It's quite expensive. So then you get this, TGN 1412. We've seen the protocol, we discussed it with the company before it was actually done. This was a study that was purely directed towards tolerability. They got the dose wrong and they found the tolerability level, but in doing so, they destroyed probably quite an interesting drug target and their own company. So this disintegrated way of dealing with drug development, non-IQ drug development, disintegrated. This is from another field. This is the what was left over from the space shuttle when it exploded, putting the pieces together here. It was actually, if you study that, a similar sort of thing. There were different parts of a very complicated organization dealing with a very complicated machine, one of these rockets that shoots space shuttles, used to shoot space shuttles up in the air. And they forgot to tell each other something, which was that there was something leaking. And the end result is a very disintegrated amount of rubbish. And I think we need to watch for that. And I think clinical pharmacologists can watch for that. And I think the place where that has to happen, and I think innovative organizations need a place like this, and I know they got one in Edinburgh and we got one in Leiden. And what I call now is not a clinical pharmacology unit. It's not a phase 1 unit. No, it should be a prototype development unit. It should be the place where the university environment develops its prototypes, because increasingly these things are also originating not anymore from drug companies but also from the university itself. We got several at the moment things that are home cooked. So a university needs a prototype development unit to teach people and to how this works. This was the Welcome Labs. And so originally you had all these different research beds in different departments, very differentiated and specialized. Everyone did his own little bit of research. It's very isolated from the clinic. Patients normally didn't get into these places. There's no contact with industry, or only in a sort of rather offhand way. A lot of the researchers bleed and feed when it was done for industry. And in fact, in most of these units, what people say, we do very boring research, so non-prototypical research, and this gives us money, and then we can do research that's fun. Of course, because of the expansion of rules and regulations and the whole thing getting more and more complicated, people didn't have time anymore to do the fun research, so they ended up just doing the non-prototypical research. And so the process for all these things comes from industry. There are the monitors that come in and they do the monitoring of the studies, and they find 1,500 queries which aren't very useful. So we're not really in a good environment there. And I think in the future, what you should get is that these places should be, and this is how we built the one in Leiden, they should have integrated in the unit both the education and the research. People should be educated to do research. You can't actually learn this when you have to do it for the first time by going into a book. It's not unlike learning to do an operation. Somebody has to show you before you can do it for the first time. And it collaborates with the clinic. It should be a place where patients can be admitted. Patients don't have to be in hospital for very long anymore. They're only there for 4 or 5 days. Whatever happens to them. So they go home, and then if they're still in a study, they must go to a place where they can actually be looked after well. The mix between internal clients for one of these units, which is your colleagues, or external ones, which often are pharmaceutical industries or non-profit organizations, is actually synergistic, so you have to accomplish that. You shouldn't separate them. It has to be operational independent. They are scientifically independent, if possible financially independent, because if they have to keep finding budget in other parts of the hospital, it becomes very difficult to have one of these core facilities. And all research should be interesting and fun. I don't believe that there should be boring research funding. Fun research, boring research just shouldn't do. And I think this will lead to publications and to a general learning process. This is what we just built in Leiden. This is the new research unit in the Bioscience Park. This is about seen from where the hospital is, about 500 meters down the road. It's made with my cousin's quadrocopter. It's a really nice toy. And this is how the inside looks. So it's an intensive care environment with monitoring facilities. But it's also got a big place where all our young researchers meet, where they can have meetings, where students meet, people who sometimes have to be away to give keynote lectures and hopefully learn something. And this is an outpatient blood-taking environment. So this was in Leiden a bit of a quantum leap in the direction of the prototyping unit. I'd like to finish with somebody whom I admired very much, Paul Janssen, the founder of Janssen Pharmaceutica, the Belgian drug company, who was particularly outspoken about how drug development went. He was a very outspoken man. And in 1983 he wrote this. He wrote about dreams. He wrote about superior drugs being, in fact, the result of a dream. And in the last sentence about bureaucrats and regulations and old prejudices and habits of mind is his words. But I think it is a vision of how, in 1983, of an extremely visionary man, how the future should look. And I think that's extremely bright. And we've written some pieces about it. I'll put this on the website so you can read them instead of listening to me, because you've done that for long enough, I think. And that was dream-driven drug development, and I wish you happy dreams. Thank you very much.
Adam, thank you very much. It's great to end on a dream. Are there any questions? Trish, if you'd like to introduce yourself, turn the microphone on.
Hello, Trish McGettigan. Thank you for that. I'm from Barton, London. And that was extremely stimulating, and I think where many of us would love to be in terms of the research and the thinking that we do. Now, how does what you said fit with the research environment-driven environments that we live in, the research returns environment that we live in universities these days? How do you balance the requirement to produce, to publish, with the dream-driven that you've just talked about?
Well, I think I'm quite hopeful about that, because we've all come from university environments that were disciplinarily organized in different departments, and we had to deal with increasingly we have to deal with interventions that are multidimensional and go across these departments. And the sort of reward mechanisms that still apply quite a lot are the ones that emphasize personal accomplishment in general. And there's things like first authorships, which we all can have quite big quarrels about. And it's actually quite strange if you think about it, because the future doesn't require that. It requires people who work together and get to a really good result. And I learned that in the ISIS group in Oxford, where when we did the big ISIS cardiovascular trials, there were no authors. It's the ISIS group. And I think that is a move, and of course there's probably no clinical trial group that's been so extraordinarily successful as they are, as Rory Collins and Richard Pito. And I think that communal approach is much more fitting with the current generation of people that are in the field. So I'm quite hopeful that that is happening, that this sort of collaborative atmosphere happens. The people who are looking after all of us, the ones that are close to their unlimited keynote lecture, still have this in their head, that we all have to become individually famous. So all hospital, university hospital executives at the end of the year at Christmas always give a lecture which goes a bit like this. I watch for them because they're quite funny. They always say, they always start by quoting how many Nature papers we had this year and who was involved in them, and they personally named them. And then they always say, boys, we have to really watch, because the research is getting more multidisciplinary and more complicated, and we have to collaborate. And the two things are, in fact, mutually exclusive. So we have to do a couple of things to get better. But I think it is happening. It's already happening. You can see these borders going. I'm quite hopeful about it.
So we have to return to the idea of a university. Thanks.
Yes.
Another question.
Goya from Vienna Clinical Pharmacology. It's just a remark, but I think at this time we need also to mention that the pharmaceutical industry, especially in Europe, has done a unique initiative to join with the European Union and really invest 2 billion in what you were talking about, this, overcome these hurdles of monodimensionalism. And I think a lot of is ongoing, that industry is collaborating with each other and is collaborating with the universities. And I must think, I was part of one of these IMI projects that really, really enthusiastic and exciting projects are ongoing. And like you said, it's happening, and it's now happening.
You're absolutely right, and I think it's driven by the outside, and I'm sure it will change quite dramatically. So.
Okay, and one last question.
Darren Austin, GlaxoSmithKline, ClinPharm. You raised the point about establishing pharmacology, proof of pharmacology in our early trials, and I think a lot of us have moved on now where we try and show that. And then you showed started with a nice plot of mechanisms in Alzheimer's disease. And Alzheimer's disease is particularly interesting because you made a comment about, well, it's good to show pharmacology, but sometimes we need to find the link to the disease itself. Alzheimer's is a particularly good example because billions have been spent. And how would you address the problem that it's pretty much a $30 million question to ask whether the pharmacology that I modulate leads to some clinical benefit? Because we've seen gamma secretase, tau secretase, we've seen the amyloid hypothesis.
No, so you do much. We do these things. So you do much more involved studies, and you do spinal fluid sampling over 36 hours, and you actually look at the pharmacological effect of the BACE inhibitor first in some area. These people don't remember much more after they've undergone that. But it requires a sort of form of integration. It's quite difficult to do. You have to, but it is all there in the environments in places like Edinburgh and the west of the Netherlands. It's all there, and it's just a question. You need to educate the patients. People have to realize that this is actually necessary for these drugs to be developed, and then it starts to work. And I think, but you could also do it differently and just give it to healthy volunteers and then do a trial in Alzheimer's and wait for 4 years and come back with a disappointing result and then discover you didn't actually block the enzyme for long enough. That is, I think, what we need to prevent. It's still possible that you block the enzyme and nothing happens.
Yeah, but one of the comments I make is that we're very good, actually. In the studies I've designed, we've been very good at showing pharmacology. But ultimately, the reason why there are so many good cancer drugs available now is we have a mechanistic link between primary pharmacology and clinical outcome. And in neurosciences, that link has failed to be established. And consequently, the experiments that are required to establish that link are multi-million-pound studies, not the small pharmacology studies.
So it's not an industry anymore that finds molecules. It's an industry that finds the disease mechanisms. And that is, I think, the move we're making at the moment.
Yeah,
that's why it's becoming fun again.
Yeah, I agree.
Okay, thank you very much. We'll end on that point. I'd like you to thank both Adam and all the other speakers for this afternoon's session. Just to end, I've got a couple of announcements, and the first is that drinks should begin to be available outside in the foyer to accompany the poster session, which is taking place all around this. Posters which are on demonstration there. And I think finally a buffet supper will be served upstairs just next to the old library as from 7 o'clock this evening for those of you who are staying in the building. Thank you very much.